Now we're going to spin down some cells in a number of different experiments. One of the first steps you have to do is to isolate your bacteria from a culture. A number of different experiments will involve whether we're the isolation of the cells or the accumulation of cells, whether we're talking about isolating DNA from cells or any other cellular component, plasmid DNA, you name it. The first step is spinning cells. So I'm going to talk about or demonstrate how to spin down cells. First, let's get some microfuge tubes. Here's our overnight culture. You can see the turbidity of the bacteria. Looks like good log phase bacteria. Micropipette, 1,000 microliters. Most of the time when you do this experiment, you'll spin down a milliliter of cells. Okay, now we're ready to spin these cells. So let's go over to the microfuge. So we have our two microfuge tubes. It's very important that your tubes be balanced. Tubes be balanced. All right. Generally, 2 minutes at 10,000 RPMs will spin down bacteria adequately and pellet those bacteria. So let's start a run. So 10,000 RPMs or 10,000 Gs with this microfuge is they're about equivalent. This isn't always true depending on the centrifuge head, and always should make sure that whether it says 10,000 RPMs and 10,000 Gs, that they're approximately the same, which it is in this case. We're going to spin for 2 minutes. I'm going to stop it now prematurely so that we can go on to the next step. So let's stop. Well, let's see how we did. There is the bacterial pellet, right there at the bottom of that supernatant. Most of the time we'll decant off or pour off the supernatant, and these are the cells that we'll work with. We'll resuspend in buffer or some other solution. I hope you can see this pellet. It isn't always visible quite as clearly, but this is pretty good.